and also to be able to handle the bioinformatics associated with that. This is something that an initiative has been taken by CAXT to deal with in Saudi Arabia, and I'll talk a little bit more about that in the next few slides. Clearly, as has been hinted by many of the presentations today, the solutions to some of these problems do in fact lie with forming associations with colleagues. For instance, it requires a cooperation of pathology, bioinformatics, and genetics, and one could argue medical informatics, to be able to create a strong next-generation sequencing program to deal with cancer. So this slide here gives you some idea of an overview of the test protocol that one would need to undertake to provide good next-generation sequencing capabilities to deal with questions arising from cancer. Essentially, one needs to collect a DNA sample. We've talked a little bit about sources of samples. To fragment that sample and to create a DNA library for sequencing. In the case of cancer, the favored option would be to create mate-paired libraries that allow you to also detect translocations in addition to insertions, deletions, or point mutations. Once the library has been created, it would go through a process of sequencing. There are several modes for sequencing. The mode that we currently use, or that we have established through CAXT, is one that uses an ion torrent sequencing capability, but the actual capability is not the important factor here. It's the volume of sequencing and or the throughput of sequencing and the time frame in which it could be done. Once sequencing has taken place, irrespective of whether that is at the genome level, exome level, or at a gene panel level, that sequence is mapped to a reference to create a BAM file which can be cleaned prior to calling variants that are present within that sample. Once those variants are called, you go into a higher level of informatics where those variants are annotated against information present in various databases. Some of these databases are for instance, the reference sequence, but also includes databases that incorporate all of the literature available associated with a particular tumor or disease. This level of the annotation level here is something that can continue to grow and can be customized to the needs of each group. Once annotated, then a report needs to be made to sift from all of those variants, the variants that are likely to be important with respect to cancer or to the specific tumor of interest. In addition to the actual test workflow, I hinted at medical informatics, and I think this is a very important part of the picture, that not only does one need to build information about the genetic variation that is present within a sample, but that needs to be associated with the clinical information. And so to collect information at the outset, together with the tumor, in terms of phenotype, pathology, etc., to take it through a process where variants are called, and then to end up with a report that goes back into an electronic health record, such that the data can be used for an individual patient, but to have that medical informatics capability where you're not only looking at an individual patient, but at groups of patients, is extremely important to leverage the capabilities of next-generation sequencing. I've just put this slide in here to highlight the fact that the infrastructure, particularly with respect to informatics, is quite considerable, expensive to put into place, and requires national or very large efforts to be able to make work properly. So it's very encouraging to see that there are several ongoing efforts to integrate molecular diagnostics into various clinical trials. This table actually comes from a very recent review in Nature Reviews in Drug Discovery, and shows many institutions that are currently engaged in using NGS within their cancer programs. Again, multiple modalities are used by these entities. Interestingly, the costs here associated with gene panels range in the high hundreds to the low $1,000 marks, right through to genomes, where the cost is about $5,000 to $20,000 currently. These costs, we believe, will come down significantly with time. Despite all of the activity in terms of identifying biomarkers, there is still a relatively modest list of markers that are clinically actionable. And I think this is the area that requires the greatest effort now in terms of leveraging next-generation sequencing in oncology. And this diagram here depicts where the bottlenecks are likely to be. What we have talked about is being able to analyze a tumor and to identify targets. Once a target is identified, to then be able to identify inhibitors, to go through early trials to work out responders, non-responders, to work out what the resistance mechanisms are, to repeat that genome analysis and then to expand the study is, I believe, the reason why the many variants that are identified within tumors through these large consortia that are currently studying many tumor types and subtypes are not immediately translating to the clinic. But once again, it would be fair to sort of say that given the explosion of biomarkers that are being identified and the clinical trials that are now underway that utilize that information, that it's only a matter of time before these biomarkers become a more regular part of clinical activity. So here I just want to quickly go through what has happened locally through the efforts of CAXT. What you see here is a diagrammatic representation of workflows that have been established and will be, I guess, announced more loudly in the coming weeks. Through the efforts of CAXT, we now have people who are in the audience who we're happy to introduce to you, who have developed a full system to deal with an NGS workflow, right from a sample registry through workflows for gene panels, for exomes and genomes, to be able to run these samples, to sequence them, to collect the data, to go through a primary compute capability, to call the variants that are present within the sample, take them through a tertiary process where variants within the sample are actually annotated against information in the databases, to report that back into the LIMS such that if additional work needs to be done, it can be recycled, and finally, to output into a knowledge database to be hosted at King Abdulaziz City for Science and Technology that will hold population variants, population variants associated with the general population, variants associated with hereditary diseases, and in the longer term, I hope, variants associated with cancer. So this has largely been facilitated, of course, by the fact that sequencing throughput has increased enormously, whilst in parallel, sequencing cost has diminished to a point where one can actually take this on. So in summary, I would like to sort of say that next generation sequencing in Saudi Arabia, through the platforms that have been developed through CAXT, are now likely to be used for disease classification, where NGS will increase accessibility for genetic testing, that greater numbers of patients can undergo screening for familial cancer syndromes. NGS can be used for molecular staging of tumors. With respect to therapeutic options, it will facilitate the development of targeted therapies and personalized medicine. It will facilitate the early detection of relapse and monitoring of residual disease. And here I think looking at circulating tumor cells through peripheral blood samples or looking at cell-free DNA may be interesting options. It will also fuel research interests in cancer by providing an opportunity for the discovery of driver mutations, for profiling genomic instability, for characterizing tumor evolution, and for looking at epigenetic changes. I think it is poised to help with target discovery in a population-specific and customized way. With that, I would like to thank CAXT for putting in the effort to help build such a capability, and I would like to thank you for your attention.
Okay, thank you very much. In the interest of time, I think we'll skip the question session, and I'm sure people will have a lot of questions during lunch. So moving on to the final talk of this session by Dr. Ashraf Talol on the methylome of breast cancer in Saudi Arabia.
Our efforts to identify the methylome of breast cancer in Saudi Arabia, and this is because we have a unique characteristics of breast cancer in the population in this country, is that the majority of patients diagnosed are usually under 50 years old. And this compares to any random study you take from an American institute, which usually find median age around the 57, 60 mark. And this could be caused by a balance between inherited susceptibility to breast cancer caused by the higher levels of consanguinity here, or also it could be epigenetic aberrations caused by increased exposure to environmental challenges, because lifestyle here changed significantly within the last 20-30 years. So at this part of the talk, I'll be representing epigenetic aberrations, but there's also a concurrent project going on where we look for the susceptibility factors. And we started by the candidate gene approach, where we selected a few genes known to be methylated frequently in breast cancer, and we can quickly see that the methylation levels of these genes are quite low in our samples. So maybe the methylome profile of our samples is quite different from Caucasian or European samples. So this led us to suggest that we need to do the methylome analysis to look at genome-wide scale. And the method I adopted, called MBD-seq, or methylbinding binding domain sequencing, basically, just a brief introduction of this technique, where you see the methylated lollipops represented by the red lollipops here, and the methylated CpGs, and unmethylated CpGs represented by the white ones. You fragment that into smaller fragments, as Dr. Brian just mentioned just now, and we prepare libraries, but before you do so, you enrich it using methyl-binding domain protein-coated beads. And these beads have very high affinity to the methylated CpGs. You wash out the unmethylated ones, and you dilute the methylated fragments. This way, you sequence it on the solid platform, and you end up with nice data regarding the methylation enrichment that you saw. This is just briefly the pipeline we utilized, because there aren't many MBD-seq specialized bioinformatic pipelines out there, so I adopted what's used for ChIP-seq, which is a similar approach, and we use the peak detection using the max version 1.4 in this case. We visualize the samples on genome browser, and we call the peaks overlapping the transcription site or a CBG island, then followed by ontology and pathway analysis. You can clearly see here that the method is validated by you pull out from a cell line here. These are enriched peaks or methylation peaks overlapping the histone regulation peaks quite nicely. And even the poorest sample here, a tumor, you can also still see some enrichment of methylation at that level. And just to show that most of the samples overlap with the transcription cell size, so we selected that one and identified those genes close to that within 1 kb of the transcription cell site, assuming that location is the one responsible for the potential silencing of the gene. We eliminated those methylation events happening in the normals, and then the cluster analysis, hierarchical cluster analysis for the methylation events represented here in the yellow. And you can clearly see from this figure that the clustering of methylation signatures show marked heterogeneity between the breast cancer samples analyzed. There isn't really good clusters originating from this kind of approach. But anyway, from the top clusters, you can find those genes, bottom and pathway analysis, and you can find that the top network of genes detected is genes affecting dermatological diseases and conditions. So that was quite interesting to see, but it cannot be in breast cancer. When the closer examination, we found that a lot of the keratin genes methylated. And that apparently because of an event happening called the long-range epithelial silencing, that usually happening at chromosome 17. But also we found a region at chromosome 12 where also there's a cluster of keratins that are methylated. So we didn't know the significance of this methylation event in breast cancer. But other genes affecting cell death, cell cycle, and cancer were also found clustering mice. But then we looked at individual genes at a time because it's easier to do follow-up functionally for these genes. One of the genes that from those methylated in the methyloma process was the melatonin receptor 1B. And that was attractive to study because especially people who live in Jeddah know how alive it is at night and how sometimes sleepy it feels during the day because the light cycle is quite different here. So we thought the melatonin receptor might be affected. And in the breast cancer itself, at least, there's enrichment of methylation at the promoter of this gene, whatever this one, here in the tumor compared to matched normals. And there's levels of methylation in the base ductal carcinoma compared to the other types, subtypes of breast cancer. Another gene also that caught my eye was leptin, which is, many of you probably recognize as the obesity gene. I thought, okay, overweight people, including me, also might suffer this. So leptin, we looked at it carefully, designed methylized assay, and also it is methylated more in tumors compared to normals. The one that really I followed is clotho methylation. Clotho is an interesting gene that is involved in vitamin D metabolism as well as could be involved in glucose metabolism and bile metabolism. So it has many functions, but what I feel is not all of it is known yet. But the knockout of this gene in mice causes premature aging. And that goes back to the original point I mentioned, which is the breast cancer samples we have, or we have here, suffer from being diagnosed quite early with breast cancer. So maybe we have some kind of a premature aging symptoms or something like that. So we looked at clotho, and you can see the methylation enrichment in the tumor versus normal, validated Douglas sequencing, and also looked at the microarray analysis we've done earlier, and the expression of that gene is downregulated significantly in most of the samples we looked at. With the assay we made for its methylation frequency, you could see that most of our samples, at least around 70% of them, have detectable clotho methylation, which makes it a quite frequent event, even higher than methylation of RASF1A, which is usually the highest methylated one. So what does clotho do? I mean, one of the things known about it is it helps to keep the kidney normal, or looking like normal. It prevents injury, or acute injury from turning into chronic injury. And the reason I'm showing this slide is just the interesting endpoint here, if clotho is not really working, is that you get the epithelial to mesenchymal transition. EMT is very common in cancer. Anybody who works with cancer heard about EMT. So maybe we have here clotho playing a part in the EMT happening in breast cancer. We don't know. So with the help of Dr. Mohammed Abu Majd here, we managed to have an antibody immunohistochemistry assay going for it, and you can easily see that clotho is expressed nicely in mammary tissues. And in some of the slides, if you look carefully, the expression is enriched towards the lumen of the ducts. So that's also these following up. But it's easier to do actually qPCR on expression analysis. So we followed the qPCR, you can see from surgically resected tumors that the expression of clotho is significantly downregulated compared to normals. And when we treat a cell line not expressing clotho with azacitidine, which reverses the methylation, you get clotho expression back quite significantly, even only after 3 days. The methylation levels are higher in the tumor compared to the normals, and it's also more specific to invasive ductal carcinoma, ductal carcinoma in situ, and lobular carcinoma, but not in the mucinous or papillary. And also the fibroadenomas do not exhibit much methylation of this gene. So what's the significance of all this? Okay, we did the statistical analysis with the clothomethylation versus all the clinical pathological parameters, and we couldn't really find much of an association. But the detection of methylation of clotho meant, okay, the whole pathway might be affected. And when you look at literature or pathway analysis, you can see clotho is actually in the center of an interesting group of genes that function mostly in metabolism. And Professor Fornes mentioned this morning, even 53, which is non-tumor suppressor gene, it can affect metabolism. So the two things are linked. So now I'm looking from the other way around, metabolism causing cancer. And you can clearly see that FGF23, FGF19, and FGFR4 might be affected. So we've got antibodies for these genes and looked at them in detail in breast cancer. And with the help of the Basil, who presented just earlier, we looked first at FGF19 in breast cancer, and actually, interesting thing was FGF19 is really strongly expressed in mammary tissues. It's very easy to detect, and 60% of the samples we looked at have high expression of FGF19. And that expression, the high expression, is significantly associated with the higher age or the older age, and with poor survival. Actually, the poor survival was strongly affected by the high FGF19. Disease-specific 5-year survival is much poorer in patients expressing high FGF19. And that effect is exaggerated by the presence of lymph node metastases or older age or negative ER. The other thing we looked at was FGFR4 expression, naturally. So what we found is FGFR4 expression correlates very nicely with FGF19. So a very strong correlation between the two. And interestingly, not just it affects poor survival, FGFR4, is that when you look at age, effect of age on the overall survival of these patients, you can see there's hardly any difference to the left here. But make that, stratify that as a function of FGFR4 expression, and you can see an interesting picture. When FGFR4 is low, the older people live better than the younger people in breast cancer. But when they have higher FGFR4, the effect is reversed, where, as you expect, the older patients would suffer worse prognosis. And so, in summary, methylome analysis using MBD-SIG approach is informative even in primary tumor samples. And what we can see, there's a marked heterogeneity between the methylomes of different samples. But most significantly, close FGF19, FGFR4, and I think FGF23 will also be shown soon, that the expression of these genes may play a significant role in breast cancer. How, the mechanism, and all these things needs to be investigated, because most of the functional studies on these genes done in liver or kidney context. And just to end by thanking these individuals, the Basil to Mohammed Abdul Majd, and the work couldn't have been done without the talent and dedication of the technical staff, Shireen Shaila, Banana Lubna, and the administrative committee of the CGMR, as well as Tawfiq and Sajjad. Thank you very much.
Okay, thank you very much. Again, in the interest of time, we'll move questions to lunch time. So it's lunch right now, and given that we're late, we can actually keep the 1 hour because it's an important part of the whole program. We're going to be back at 2:15 for the keynote lecture, so please, everybody be here by 2:15. Okay? Let's thank all the speakers again for this session.